It is my request to communicate the presence. Kindly communicate the presence. Sorry because I am late today by 10 minutes. Can my sound is audible? Kindly confirm that I am audible to all. Kindly communicate. Am I audible? Yes, okay, okay. Now, we are going to discuss on the Hardy-Weinberg law principle. And from the Mendelian principle, we observe that the alleles are present in two forms. One is dominant and one is recessive. So, if we consider the allelic frequency, then it is possibility to observe more dominant characters than the recessive characters. But it is not the truth. Sometimes we observe the more frequency of the recessive genes, and the question was at first asked to Batson when he was delivered a lecture in international symposium, and after the returning from the symposium, Batson communicated the view to his two friends. One is Hardy and one is Weinberg, and both were experts of the mathematical analysis. And when they calculated the frequency of the first generation uh, and compare it from the subsequent generations, both independently observed that the frequency is always constant when the evolutionary and environmental factors operated on particular gene is negligible or zero. And after that, we know that the Hardy-Weinberg law is existed. If we consider alternative of the Hardy-Weinberg law, then we are easily say that the change of the allelic frequency in any population from his his parental stock represents that some particular environmental or evolutionary factor must be operative on that particular locus. In this way, the Hardy-Weinberg law provides a basis for the research and analysis of the gene equilibrium and provide a base for the analysis of evolutionary effects of environmental factors. Here, the topic is most important both for the postgraduate and NET examinations. This year also, one question is asked from this part, and the questions are always asked from the Hardy-Weinberg law principles regularly in the competitive examinations. And the factors which are responsible for the change of allelic frequency are selection, mutation, drift, and migration. These four factors are commonly observed for the and for the changes in the allelic frequency or gene frequency. So, the two concepts are, I am just explaining here, the two concepts are commonly considered for the study of the Hardy-Weinberg law. One is the gene pool and another is the gene frequency. The gene pool represents the total number of allelic components present in any population or the total number of genes present in any population. Other way, the gene frequency represents the percentage prevalence of gene at any particular locus in any population. So, the if we consider just I am giving an example, and if we consider that the five individuals, if the five individuals uh, are studied in which two represented both dominant genes and three represented both recessive genes, because the genes are present in pair in allelic components, that is they are homozygous, or if both are present then it is referred as heterozygous. So the in this condition, the gene frequency is represented as P plus Q. And if we mention the allelic prevalence in on the basis of the appearance of the phenotypic character, so the the total value of the total value of the gene, dominant and recessive gene, are equal to 1 probability. If you consider the probability value, then the P plus Q plus 1, P plus Q equal to 1. That means here in any population, the if we observe the phenotypic character only, then it is difficult to say that the dominant character is present in homozygous condition or heterozygous condition. So, when you analyze to calculate the gene frequency of dominant and recessive gene, then we fundamentally or primarily consider the recessive characters observed in any population, because the recessive characters are always present in homozygous condition. There is no chance of the presence of recessive character in hemizygous condition or heterozygous condition. So the dominant gene, if we observe the dominant gene or represented the dominant gene by A and observed in 50 characters, 50 individuals, so there is chance that among the 50 some are present in AA configuration and some are present in capital A and a small a combination. In this condition, when we observe the phenotypic character in any population for any allele, then it is very difficult to say that which person is present in capital A combination and which individual is present in capital A small a character. So we always calculate the gene frequency or allelic frequency in the form of recessive character, and the recessive character is represented by a small a in my example. So the if we observe the 10 recessive individuals, it is clear that the mm, individual individuals are in the combination of AA. So the A square, sorry, Q square is Q, that is the recessive character. Q represents the recessive character in the Hardy-Weinberg law principle. So Q square is always represented by mm, 10 divided by 60, because 50 individuals represented dominant character and 10 individuals represented recessive character. In this condition, the Q square represented the value Q 10 minus 60, that is 1 by 6. That is the value of the q square. And when we calculate the root value of the q, if we want to calculate the q, then the root value of the 10 minus 60 is represented the probability value of the gene frequency of recessive character, or character of the recessive gene. Suppose we is here further suppose that specifically the Hardy-Weinberg law states that at in a large, what is the Hardy-Weinberg principle? I am just saying the Hardy-Weinberg law principle, and it states that in a large uh, randomly mating populations, or the populations has uh, panmixis, and if the population is large and has randomly mating with a closed gene pool, because if the gene migration or gene flow is absent, so these are some conditions for the maintenance of Hardy-Weinberg law. So we restrict, or we provide some conditions where the Hardy-Weinberg law is applicable. And the Hardy-Weinberg law states in a large randomly mating population with a closed gene pool, the gene frequency remains constant from generation to generation. It is the Hardy-Weinberg law. And if the gene frequency is changed, then it represents, alternatively we consider, and it represents that some environmental and evolutionary factor must be dominated on that particular locus. So we consider here to explain the Hardy-Weinberg law, and let consider that P is the frequency of the dominant allele, B, capital B. We suppose that the P is the frequency of the dominant allele, capital B, and Q is the frequency of the recessive allele, small b. We suppose here. Then all gene frequencies of alleles of a gene, that is P plus Q equal to 1. a propagation of the individuals. Of course, it is the parent stock, and suppose a propagation of the individuals has each possible genotype. Each genotype has a frequency in the population, and the frequency of the all three genotypes must add up to 1. So, if the P plus Q is equal to 1 and we square both left and right hand side, then P plus Q square represents P square plus 2PQ plus Q square as messaged by the Nabia Raj in the my this live is equal to 1 square, that is 1. So it is extended in the form of P square, that is PP. That means both capital, both dominant characters are present here, plus 2PQ square, that means 2 dominant and recessive, that is the heterozygous condition, plus 2Q square, that is both recessive QQ conditions. So here the expanded part of the Hardy-Weinberg law. And here the 1, the P plus Q plus 1 represents the phenotypic nature, whereas the P square plus 2PQ plus Q square represents genotypic nature of the population. So here, if we consider, because we consider that the dominant is represented by capital B and recessive is represented by a small b, so we write here that frequency of capital B plus 2 frequency of capital B small b plus frequency of BB, that is equal to small b, that is equal to 1. If we converted the P square plus 2PQ plus Q square is equal to 1 into the actual nature, because we consider here the capital B as the dominant and small b as the recessive, so the formula is converted, the explanation of the Hardy-Weinberg law is converted into BB plus 2 small b capital B plus small b, that is equal to 1. The Hardy-Weinberg law predicts that no matter with what initial frequencies of these three genotypes the population begins, in one generation of random breeding, I am saying that one generation of random breeding, because in my class a student asked that when some population is removed from the main stock or parental stock and represented the population migration from that particular geographical area, then what will happen? Can the gene frequency is changed? So I said in my class that no, there is no change is observed in the parental stock. Here, here the explanation. Just I am saying that the Hardy-Weinberg law predicts that no matter with what initial frequency of these three genotypes the population begins, in one generation of random breeding, the frequencies will be given by the binomial expression of P plus Q square. That is P square plus 2PQ plus Q square. And they will be remain constant thereafter. And after that, the frequency which is existed after the first random mating will be maintained from generation to generation, and it is the Hardy-Weinberg law. And again, we use some numerical points for the explanation of the Hardy-Weinberg law, and I suppose that you patiently uh, observe the facts, or it is better to write if you have pen and copy. Then the uh, in the uh, observe the Punnett square and see the one frequency, the frequency of dominant is zero point 0.6. The frequency of dominant gene is zero point 0.6, and the frequency of recessive gene, that means, is the 0.4. And in this condition, when we multiply and when we observe the p square value, that means the 0.6x square, that is 36, and here p square represents 36. When we observe that the p value, the frequency of the dominant gene is zero point 0.6, then the homozygous dominant populations are 36 in 100. I am just asking in the 100, or in the probability it is 0.36. In heterozygous condition, the dominant in the heterozygous condition is present in the form of 2q. That means 2 multiplied by 0.6, again multiplied by 0.4, and the value becomes 48, 0.6 and 0.4, 48. That is the 48. And here, the if we consider the value of q square, that is 0.4, is becomes the 0.16. That means if we calculate the 36, 16, and 48 becomes the 1 or 100, and if you convert in the probability, it becomes 1. So it always remains the comes in the term of 1. So for the first generation of progeny, the genotype frequencies are given by the relationship p plus q square, that is p square plus 2pq plus q square. In this condition, the Hardy-Weinberg law is explained by the binomial expression of the p and q. In further, if we moves in the next generation, so there is chance of 3.6, that is 36, 36 dominant character, and that means the 0.6, that is 36, is pure dominant, that is represented by capital B and a small b, and 0.4, just you try to understand what I am saying here, that 0.6 square, that is 36, has pure, not habit, pure dominant, and 0.4 square, that is 16, is recessive. So there is a chance of probability to multiply to twice by the pure dominant and pure recessive. One, if you represent the diagrammatic, then 0.6 multiplied by zero point 0.4, again 0.6 multiplied by zero point 0.4. So if you try to randomize the relationship, so 2PQ is conformed. So here again we calculate the frequency after the next generation, it again represents the same ratio. So when we move from generation to generation, the uh, allelic frequency or gene frequency at any particular locus remains the same, and it is presented by the Hardy-Weinberg law. Now the conditions represented, the suppose the another example, and here we consider the phenotype of a dominant character. The phenotype of a dominant character appear in 36 individuals. If we go and do any work in field and considered, you observe the dominant character in 36 individuals out of 100. That means the 36%, and obtain that the gene frequency, what is the gene frequency. So here we observe the dominant character, not the recessive character. I am asking here that you go in the field, it is better to understand what I am saying, that you go in a field and observe the 36 dominant character in out of 100 individuals. So we observe the dominant character in the 36 population, and it is not possible to calculate the gene frequency or genotypic frequency on the basis of the dominant, because the dominant may be present in the homozygous condition or heterozygous condition. So by the use of formula P minus Q is equal to 1, at first you calculate the value of Q. And here the Q is, if the 36 is represented in the 100, then it becomes 0.36 if we considered in the probability scale. So the 0.36 plus Q is equal to 1. And then you observe the Q is 0.36 minus, 1 minus 0.36. So 0.64 is the 0.64 is the number or the frequency of the recessive gene. Again here the 0.64 in 100 it is the 64, but in probability it is 0.64, and then the 0.64 represents the Q square, because Q is present in QQ form, that is the Q square. And so the Q is represented by or is calculated by under root of 0.64. That is, it is calculated as the 0.8. And here that means the frequency of recessive gene here is 0.8, and the frequency of dominant gene is here 0.2. And when you want to calculate the nature of the different genotypic individuals in the term of probability or in the term of A, so the 2 square, 0.2 square, that is the 0.04 is represented by P square. The value of the P square is 0.04, that is the square value of 0.2. And when we consider the value of the 2pq, then it becomes 0.32, 0.32. And when you add the 0.32 plus 0.04, it becomes 0.36, as we observed in the in your survey. So you observe the 36 individuals representing the dominant nature, but the gene frequency, if you calculate the gene frequency, it becomes zero point 0.2 for the dominant gene. And now q square, if you represent the q square, that means the individuals having the recessive gene, that is 64. So here it becomes clear that you always calculate the gene frequency for the analysis of the Hardy-Weinberg law principle by the recessive character, not by the dominant character. Again, I am saying that the dominant character represents both homozygous and heterozygous conditions. So the actual value of the homozygotes is less than the individuals representing the dominant feature in any population. Now, we consider here, we discussed here, and as I suppose that we understand properly that what is the method of the calculation of the gene frequency and analysis of the Hardy-Weinberg law principle. Here again, it is better to understand that a population in which a constant ratio of genotype is seen to persist from generation to generation is referred as genetic equilibrium, is referred as that the population is present in genetic equilibrium for that particular gene, not for the other. The gene or the allelic combination which is analyzed, if represent the persistent constant gene frequency from generation to generation, is represented the genetic equilibrium. The status of another gene need to identical, however, if the genotypic frequency for a gene are not constant from generation to next generation, the gene is said to be present in genetic disequilibrium state. And the disequilibrium state of the genetic status in any population caused by a violation of Hardy-Weinberg law assumptions. And the gene frequency are changing due to selection. Just I am saying that gene frequencies are changed due to operation of some ecological and environmental factors on that particular locus. That may be selection, may be migration, may be mutation, may be non-random mating, may be selective marriage, may be meiotic drive, like this. There are a number of factors, or gene flow, genetic drift. There are a number of factors which are responsible for the changing of the gene equilibrium. So the consider here the one gene, one allelic pair, but sometimes we observe that the one locus is responsible for three three genes, or it may be said that the locus is multi-allele, multiple allele. In this condition, in population, it is 100% when all the multiple frequency of all the multiple alleles are added. Then suppose a locus has A, B, and C multiple alleles. So the frequency of A plus frequency of B plus frequency of C is responsible for the 100% expressiveness of that particular locus. In this condition, we said that the allelic nature becomes P plus Q plus R equal to 100%. As we observed in the condition of blood. But here in blood, one thing extra is appeared, that is the codominance. Because the A and B are codominant, whereas R is recessive to A and R is also recessive to B. So here the multiple allele and codominance are also present. So the calculation for the frequencies of the multiple allele or for the codominance, the gene locus has the multiple allelic with the codominance are changed, and that we will discuss in further class. And here the components, the third component is the character or the allele which is associated with the sex chromosome, that is the X chromosome. And here you simply understand that the frequency is always calculated in term of the total number of locus present in any population. In the autosomal linked alleles, it is well known that both male and female have equal number of genes locus site. So it is, if it is multiplied, but in the condition of sex linked gene, the male represents variation from the female. The female behaves same as the autosomal conditions, but in male the gene behaves not as the autosomal gene, because in male only one X chromosome is present. So the presence of gene is expressive and always present in pure. There is no chance of the heterozygosity of the X-linked recessive gene in male. So in male, if the sex-linked frequency is asked in your question, then in male, the X-linked recessive gene represents P plus Q is equal to 1 always, both in phenotypic and both in genotypic nature. There is no need to square it or calculated by the use of under root. But when the chromosome, the X-linked gene is X-linked recessive gene is present in the condition of female, it follows as P square plus 2PQ plus Q square. So here the nature for the calculation of the allelic frequency as per the Hardy-Weinberg law is different both in male and female. So as I consider that you all properly understand the Hardy-Weinberg law and the method of calculation of gene frequency as per the principle of Hardy-Weinberg law. Now, I am thankful to you all, and you are free to ask or give me message for any question. If you have any problem, because I am delivered a short lecture today, just after the 42 to 45 minutes, because it is some new for you, and next day I will extend it, probably tomorrow. Okay, thank you. No question is asked. Okay, thank you to you. Thank you all.